Okay, so you should have the new questions that I redid with you guys because somehow I got that saltwater one. That's an old one, so everybody should have the new one. And I fixed it in Canvas too. So, alright, so let's look at your chart. Alright, let's review over the different types of solutions first. So you have hypertonic. What should that one do? You've got, remember
it
you've
should
got
increase.
hyper and hypo.
It should increase, right?
Hypotonic, the O means that it swells.
Yeah. Oh, okay.
So hyper means that it should do what?
Shrink.
It should lose mass. Hypotonic should swell, so it should gain mass. And then isotonic should stay the same, in theory. Okay? That does not necessarily mean that those are results that we're going to get. One, I don't know the concentration of stuff in the grape, so I was just kind of guessing with the isotonic solution using the juice. And then, I mean, the solutions that I made up, they're just kind of estimates based off of what the grape should have in it. So, did anybody's hypertonic shrink? Did it lose mass?
No.
Yours did?
Pretty sure mine did. Yeah, mine lost mass.
Oh wait, I think I measured mine wrong. I think one of them lost a gram and one of them gained a gram.
Okay.
Yeah, one of mine stayed the same, one of mine swelled up, and one of mine shrunk.
Okay, so Hayden, what was your hypertonic mass before?
2.
2 grams. And what was it after?
3.
Hypertonic?
Oh, I did it wrong, I guess.
Yes.
Oh, that one is 3.
This one went from 2 to 3? That was your hypertonic?
I don't know what I did.
Take off your glasses.
Brady, what'd you get?
I had for first I got 3 grams, and then after I got 2 grams.
Okay. Alright, so that one we saw that did it lose or gain? So it lost mass. It lost 1 gram. Okay, whose juice stayed the same?
No.
Mine did
Yours did? Alright, Michaela, what did you get for
6 grams and 6 grams.
6 grams, 6 grams, so we're going to put same out here. Whose hypotonic solution actually gained mass?
Okay, now I'm here.
Okay, now we're on it. Alright, Malachi, what did you get?
5 grams before.
Okay.
And 6 grams after.
Okay, so this gained. Alright, so you do not have to write these up here. This is what I'm going to give the people who are remote. So this is just kind of a combination of ours. So theirs, in theory, is going to work out perfectly, where ours didn't exactly work out perfectly. Some of you, yours stayed the same. Some of you, it gained or lost. And that's totally typical in science. That's why you have to run so many trial runs of the stuff that's going on. In an actual science laboratory, we would have taken the composition, the sugar composition of the grape, and try to get these based off of that. Okay, so, but we're just running on guesstimates. But for the most part, we got what we were supposed to. Okay, so hypertonic, it shrunk for a lot of people. Isotonic stayed the same. Most people definitely saw this hypotonic, right? Most people definitely did that. That's because we put a grape in tap water. So in theory, the tap water should not have had sugar in it. And so the grape does. So that means that the water rushed from the fresh water to where it was real sugary, and it made it swell. So that's why we're seeing a lot of that. These other two, that's why it's kind of hit or miss. It's based off of the grape, it's based off the composition of it, and if I got the solution right. Okay. We might have also seen a different mass with them if we would have given it a little more time. Sometimes this takes a little longer. We've given it what, a couple days?
Yeah.
So that's not really a long enough trial to see really, really good results, but it's good enough for what we want. Okay? So let's look at the questions that you got to answer. So the first one's explain what happened to each grape in the experiment. Use the terms osmosis, semi-permeable membrane, concentration, and either the following: hypotonic, isotonic, or hypertonic. So that will be based off of your chart stuff. What is this semi-permeable membrane? What does that mean?
It lets certain things in and out.
Yeah, it lets certain things in and out. We're talking about osmosis here, and what is this substance that's moving in and out with osmosis? Water. Okay. All right, so you can go back and answer that one in a minute. The next one say, what could be a potential source of error for this experiment? I've given you a bunch of different reasons why it could have messed up. Okay, so any of those that I've already talked about. And then the next ones, and it's okay if you have to look these up, or it's okay if you think about them, okay, and just come up with an answer with it. Explain how osmosis is involved in the following: a grocery store spraying water on fresh produce. Okay, so you're putting fresh water on it. Basically, you're asking what's it doing. This goes back to the wilting stuff I was talking about the other day. Remember, osmotic pressure, it absorbs water and it makes it what? We're talking about a plant. If you've got a wilted plant, what happens when it absorbs water?
It'll look younger or healthier.
Yeah, it looks healthier. It perks up, it stands back up. That's osmotic pressure. It took in water, and then the structure of the cells in the plant, remember they don't have a skeleton, they have a what?
Cell wall.
Yeah.
Cell wall, so that helps it stand up. A person getting wrinkly in a pool. If you're getting wrinkly in a pool, is water coming in or out?
It's going out.
Out, so that would be hypertonic. It's making your cells shrink. Putting salt on a slug.
Kills it.
Yeah, well, it does kill it.
Because water goes out.
Water is being pulled out of the slug, so it's making its cells shrink. A person drinking salt water and becoming dehydrated. If you're taking in salt water, it's really highly concentrated with salt as opposed to your body. So the water's going to go from your cells to the salt water, and then it's going to dehydrate you like that. Preserving meat by salt curing is the same sort of concept. We talked about this one the other day, placing a saltwater fish in fresh water. So we talked about what that would do the other day. Would water be moving in or out with that?
Water would be moving.
In it. So what would the fish look like?
Blow it.
Yeah, it would like blow
It's like a
up,
blobfish.
swell up. Saline solutions for IV medication. So saline is just another word for what?
Long.
Yeah, salt water. Most of the time when they give you an IV, they put it in a saline solution. A lot of your shots are saline solutions that just is, first of all, it helps with absorption, and second of all, it doesn't burn as bad. A saline-based shot does not burn as bad as a water-based shot. Soaking potatoes in salt water before frying to make them crispy. So if you're soaking a potato in salt water, what is it doing?
I'm not sure, but I know when you
Look,
do.
I'll think about it. You're soaking it in salt water.
Just marinate.
The water in the potato is going to do what?
It's gonna exit it, right?
Come
I was
out.
about to say, doesn't that happen with like bacon and grease? Doesn't it shrivel up because of the?
Yeah, because it's
So
losing
it's
stuff.
like the same concept.
A kidney dialysis machine. So you're going to have to look this one up. This is one I'm going to make you look up. And see how that works and explain it in terms of osmosis. Soaking vegetables in water to crisp them. That's what we've been talking about with plants. And then the last part, explain why osmosis is an important factor in cell biology. You also, on that chart, underneath it says draw a bar diagram. Don't worry about doing that. Just answer your questions, okay? Once you finish this, then we'll take a look at the photosynthesis stuff. I think I'm gonna let it run a little bit longer, like over the weekend. You can see that some of them are producing bubbles. Some of them are tightly packed in there. The bubbles can't really move up real well. So what we may do when you finish these, if yours is one of the ones that's tightly packed, I may get you to try to loosen it up a little bit. Like take one of those stern rods and push the stuff over so the bubbles can move around, okay? But finish your questions and then we'll look at those.